The program lists Lenin Perez as the next speaker. Unfortunately, Lenin is ill and couldn't be with us today. So very kindly, Benyam Kinde, sorry, Benyam Kinde has agreed to deliver a talk instead of Lenin. Okay, so Benyam, please come and talk about the, well, Benyam is working with Michael Summers in the HHMI lab, and his title is Towards the 3D Structure of the 198 nucleotide core encapsidation signal of the dimeric genome of Moloney murine leukemia virus, nuclear magnetic resonance investigation of the SLB duplex.
Good morning. As Dr. Lindell just said, my name is Benyam Kinde. I guess a more simplified title you'll see on this title slide. Structural insights into how retroviruses package their genome. So as the title in the program suggests, we work with the Moloney murine leukemia virus, which is a model retrovirus. And let's first take a look at the generalized life cycle of retrovirus. So So as you can see, the life cycle is really divided into an early and a late phase. Early phase is really defined by infectivity, where the virus particle binds to the membrane, its genome is integrated into the host cell. In the late phase, more specifically, this region here, which we'll be focusing on, is defined right after proviral DNA has been reverse transcribed into RNA. Two dimeric copies of this dimeric RNA come together, causing a conformational change, which allows this NC region of this GAG polyprotein to recognize and bind this genomic RNA and take it to the membrane for assembly, budding, and eventual maturation of the virus particle. So to reiterate the part that we'll be focusing on, the two identical genomic RNA strands come together and dimerize, which allows NC to recognize and bind, and the GAG polyprotein direct the dimeric genome to the cell membrane for packaging to occur. So why study the Moloney murine leukemia virus? There's a lot of other retroviruses, such as HIV. Well, first, it serves as a good model retrovirus for other retroviruses. This is namely because most of the proteins that are encoded by the genome are specific and core for this replication process. Don't really have too many other accessory purposes. In addition, most of the virology and biochemistry of the Moloney murine leukemia virus is well understood, and it's already used extensively in gene therapy trials. So, as I mentioned earlier, we're really mainly focused in the three-dimensional structure of the core encapsidation site, which is shown here in its monomeric form and here in its dimeric form, highlighted in blue. In its monomeric form, it consists of 3 stem loops B, C, and D. And in its dimeric form, SLB interconverts into a helical duplex, and C and D maintain their hairpin structure but cross-kiss. So for the purposes of this talk, we'll just be focusing on the B duplex and the structure it takes in this dimeric form. So why is dimerization necessary in the first place for packaging to occur? Well, if we take a look at the monomeric version of this core encapsidation site, we see these red regions, which are known to bind NC of that GAG polyprotein, are sequestered in base pairing. So when NC is added, NC is unable to bind, and we're led to believe that little to no packaging can occur. However, in dimeric form, a conformational change occurs, allowing these once base sequestered NC binding sites to be free and able to bind NC. So when NC is added, NC is able to bind, which leads us to believe that packaging can continue. So project goals were first to verify that the SLB duplex forms a stable homodimer in isolation, and determine 3D structure of the SLB duplex using both local restraints and global restraints. So beginning with sample preparation, SLB was prepared via in vitro transcription by the T7 RNA polymerase enzyme. After isolation and purification, the B duplex is dissolved in an appropriate buffer and placed in the NMR. The NMR, or nuclear magnetic resonance, utilizes a strong magnetic field and radio frequency pulses to gain structural insight into macromolecules such as RNA. So the data that we collected can really be defined into two ways, both local restraints and global restraints. First, let's talk about global restraints, which can really be determined by the NOESY, or nuclear Overhauser effect spectroscopy spectrum. We'll talk about global restraints a little bit later, which are determined by the heteronuclear experiment, the HSQC spectrum. So the NOESY spectrum, it shows interactions between protons within nucleotides and with surrounding nucleotides less than or equal to 6 angstroms apart in space. Interactions occur because of dipole-dipole interactions or through space interactions as opposed to through bond interactions. And cross-peaks occur interactions occur as on a cross-peak intensity map, which we'll see next. And so by using the secondary structure, we can find a pattern in these cross-peaks and assign all the peaks in the spectrum. So if we take a look at a predicted secondary structure of the B duplex, and we take this highlighted region here for illustrative purposes, we can see how these proton-proton interactions occur. So as I mentioned, protons can interact with protons within nucleotides. So the H1 prime proton can interact with the H8 proton within the G290 base, as shown here. Protons can also interact with protons with surrounding nucleotides, as the H8 proton of G290 can interact with the H1 prime proton of A289. So it's by this method we can go through and determine all the predicted proton-proton interactions that we expect to see in our NOESY spectrum. So the NOESY spectrum shown here, on the horizontal axis we have our H6 and H8 proton frequencies, and on a vertical axis we have our H1 prime proton frequencies. In reality we have other frequencies, but this is just to go with the simplified secondary structure. So as I mentioned in the previous slide, H1 prime and H8 of G290 shown in interaction. So it's known to show an interaction at this point here. So what this tells us is that on the same vertical line, or on the same horizontal line, we know all cross-peaks that will show an interaction here are associated with the G290 H1 prime. And on the same vertical line, we see that all interactions that occur on the same vertical line show an interaction with the H8 of G290. So by this method, we can make the next step in our secondary structure in assigning the spectrum, which logically would be a vertical step down. And likewise, we know that A289 H1 prime proton frequency occurs here. So from there, we can also make the next jump in our secondary structure in assigning our NOESY spectrum, which would be a straight horizontal step. So it's by this method we can systematically go through and assign all the peaks in our NOESY spectrum. Now, as I mentioned before, this is a homonuclear experiment, so it correlates proton to proton here. Here's a assigned NOESY spectrum, and I should also mention that proton size can be correlated to the distance between two protons in space. So the farther apart two protons are in space, the smaller the cross-peak, and the greater, the smaller the distance between two protons, the greater the cross-peak. So to reiterate, the project goals were to first verify that SLB formed a stable duplex in isolation, and second to determine the 3D structure of the SLB duplex using both local restraints and global restraints. So we determined that the SLB formed a stable duplex in isolation by by making those straight horizontal and vertical steps in our homonuclear nosy spectrum. And we also obtain local restraints. Now, using those local restraints, we can obtain global restraints and a further step towards a three-dimensional structure. So, obtaining global restraints. So by measuring the B duplex in various experiments where our sample is in a aligned media such as phage, which kind of keeps the sample in a quasi-solid state or a preferred orientation, we can gain insights into physical parameters known as RDCs or residual dipole couplings. So here's an HSQC. This differs from our nosy spectrum in that instead of relating proton to proton, now we're relating proton to C13. So using our two-dimensional nosy spectrum previously, we're able to assign these peaks specific C13 proton interactions. So as I mentioned, we measure the sample in an aligned media such as phage and an unaligned media. So the sample RNA is then able to take a preferred orientation. And when we compare these two spectra, what we see is a shift. This shift is, if we take this A289 or A297 as an example, this shift can be related to a frequency because the measure in these cross peaks is frequency. So it can be related to carbon-hydrogen vector angles. So this is an example of the A nucleotide here and the C2 interacting with the H2. So as a result, when we obtain many carbon-hydrogen vector angles, we can then gain insight into other physical parameters such as RDCs. And when we have a lot of these, we're able to relate one end of this B duplex relative to the other. So by this method, we can obtain high-resolution structure of RNA. And in this case, it was the B duplex. So as I mentioned, the B duplex is just one part of the core encapsidation site, which is 198 nucleotides. And at such a large size, by NMR standards, it's hard to determine high-resolution structure. So by kind of using a divide and conquer method, we can structure the B duplex and then see its part in the entire BCD complex. So this was the first stepping stone. So in conclusion, S will be formed a stable helical duplex in isolation, and local and global orientational restraints were obtained. But probably more importantly, we established a protocol for the high-resolution structure of the entire core encapsidation site. So now we're at a position where we're able to determine the high-resolution structure of the entire core encapsidation site by this protocol that we established, which we're currently underway. And this would be significant because it will be the first high-resolution structure of the core encapsidation site by NMR, and it will also be the largest nucleic acid ever solved by NMR by these high-resolution techniques. So I'd like to acknowledge Dr. Summers, Dr. Blanton Tolbert, Dr. Yasu Miyazaki, Tyre Summers Lab, and the Meyerhoff program. And this is the RNA group that we have in the Summers Lab. Thank you for your time. Yeah, exactly. So a lot of the patterns that we see in the B duplex, we can assign in the presence of the entire BCD complex.
Is there any evidence or any theoretical evidence even that other parts of the 198 nucleotide complex will invade that helix once you have the B duplex and the context of the whole thing?
No. So the B duplex actually forms a stable helical duplex in isolation, and also what we found in relation to the entire complex. It maintains most of the cross peaks that we found in just the B duplex. Yeah.
What are some of the challenges of solving a really large structure by NMR?
Well, first, the the molecule, since it's so large, tumbles a lot slower in solution. So as a result, you get, when you're obtaining your spectrum, broader peaks, which in essence just leads to lower resolution of the structure. And it gives you difficulty when you're actually trying to assign specific peak proton-carbon or proton-proton interactions.
Thank